Hello everybody, in this video lecture we will cover biomolecules. So this is a PowerPoint that I will use to cover this material. Let's go ahead and begin. So I need to pick up the pointer. Okay, the large molecules necessary for life that are built from smaller organic molecules are called biological macromolecules. There are four major classes of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acid. And if you can see over here on this diagram, you are giving the examples of these macromolecules and where you can find them. Let's say carbohydrates used for store energy, functional materials. So you can find it in lots of food that you eat. Lipids for store energy. Proteins have variety of function, including enzymes, structural molecules. Let's say your hair is made of keratin, that is type of structural protein. Nucleic acid, example is your DNA, store genetic information. Biological macromolecules are organic, meaning that they contain carbon and hydrogen. So organic molecules are made from carbon and hydrogen and some other atoms. In addition, they may contain oxygen, nitrogen, phosphorus, sulfur, and additional minor elements. Carbon forms large, complex, diverse molecules necessary for life's function. So on this diagram, you can see here's a carbon atom that connected to 4 atoms of hydrogen. These lines represent covalent bonds, and this is the simplest hydrocarbon that is called methane. Right? But carbon can form chains of carbon. They vary in length. Sometimes double bond can form between carbon, and the number of double bonds can vary. Carbon skeleton also can be unbranched or branched, and it can be arranged in rings. So carbohydrates is our first group of macromolecules, organic macromolecules. Carbohydrates can be represented by the formula carbon, H2O, and time. That means that in carbohydrates, you have as many atoms of carbon as you have atom of oxygen, and you have twice the amount of hydrogen atoms. Or you can say it's a carbon and water. That's why it's carbohydrates. So ratio of carbon to hydrogen to oxygen is always 1 to 2 to 1. And N represent number of carbon atoms in the molecule. So based on how many these carbon atoms we have, we can classify carbohydrates into 3 subtypes: monosaccharides, disaccharides, and polysaccharides. Example of monosaccharides include glucose, fructose, galactose, ribose, deoxyribose, that's also monosaccharides. Disaccharides, maltose, sucrose, lactose found in milk, so it's a milk sugar. Sucrose is your table sugar, the one that you use in your coffee in the morning. Maltose, it's a grain sugar, you will find it in beer, for example. Polysaccharides include starch, glycogen, cellulose. Starch, you find lots of starch in food such as French fries, right? Potato rich in starch. Glycogen, only found in animals. Starch, by the way, only found in plants. Cellulose is another type of polysaccharide, as people cannot digest cellulose, and it's also found in plants. So monosaccharides, mono means one, saccharide means sweet. Those are simple sugars, the most common of which is glucose. The number of carbon atoms usually range from 3 to 6. The names end with the suffix "-os", like over here. glucose, maltose, fructose, sucrose. So you see this suffix "ose," that means sugar. Depending on the number of carbon atoms in the sugar, they may be known as trioses, 3 carbon atoms, pentoses, hexoses, 5 or 6 carbon atoms. And as always, carbohydrates, we have carbon ratio to hydrogen and water 1 to 2 to 1. And this is formula for glucose. C6H12O6. So monosaccharides exist as a linear chain or as ring-shaped molecule in aqueous solution. So in aqueous solution in water, you will find monosaccharide in the form of this ring. Glucose is an important source of energy. Galactose, part of lactose or milk sugar, and fructose, found in fruit, are the common monosaccharide. So if you look over here, glucose, galactose, and fructose, they all have the same chemical formula, C6H12O6, but they have different structure, and this is known as isomers. Disaccharides, di means 2. Disaccharides form when 2 monosaccharides undergo dehydration synthesis reaction. Dehydration synthesis removes equivalent of a water molecule to link molecular unit. Dehydration means you're losing water or you remove water molecule. Dehydration synthesis requires energy and builds macromolecules from smaller subunits. So over here you see the short polymer and the monomer that they combine together through the formation of the covalent bond, and water is removed. So we're synthesizing a larger molecule. That's why it's a reaction of synthesis. And water is removed. That's why we call it dehydration synthesis. And this reaction requires energy. Hydrolysis adds equivalent of a water molecule to break apart macromolecules, and hydrolysis releases energy. So they kind of like opposite. So here you can see a longer polymer with the water added. This polymer is split into 2 smaller molecules, right? That's why it's hydrolysis. Lysis means you break something apart. Hydrolysis, you break it apart with water added. So this is how disaccharides and polysaccharides can be formed, and this is how they can be hydrolyzed. So disaccharides, common disaccharides include lactose in milk, maltose in beer, malted milk shake, and malted milk ball candy, and sucrose and table sugar. And here's the structural formula. Oh, it's kind of like, it just show you the structure. It's not really a formula, but sucrose, table sugar, is made from glucose and fructose. Lactose in milk, glucose and galactose. And maltose in beer, glucose and glucose. So it depends what molecules are connected, right? We get different disaccharides. Polysaccharides, poly means many, a long chain of monosaccharides linked by covalent bonds. Starch and glycogen are examples. Starch is stored form of sugar in plants, and it's made of amylose and amylopectin. That's both polymers of glucose, right? So both are pretty much made of glucose. And glycogen is a storage form of glucose in humans and other vertebrates, and it's made of monomers of glucose. Glycogen is the animal equivalent of starch. So plants and animals, they store sugar in the form of polysaccharides. So plants store sugar in the form of starch, and animals vertebrates store sugar in the form of glycogen. So here on this diagram, you can see starch, how it's made from this, glucose molecules, and glycogen pretty much made from glucose as well, but different structure. Cellulose is one of the most abundant natural biopolymers. Cellulose is also polysaccharide. The cell walls of plants are mostly made of cellulose, which provides structural support to cell. Wood and paper are mostly cellulosic in nature. Cellulose is made up of glucose monomers. The glucose-glucose bonds in cellulose cannot be broken down by human digestive enzyme. That means you cannot digest cellulose. So when you eat a lot of veggies, right, they do have lots of cellulose. Let's say you eat lettuce, right, or cabbage. Pretty much all the plant products that you eat, they have lots of cellulose in their cell wall, and in our diet we call it fibers. Even we cannot digest it, fibers are good for your digestive system. Chitin is another example of polysaccharide. Chitin, well, if you ever stepped on a cockroach and you hear this kind of like scratch, that's the exoskeleton of many of these animals. It's made from chitin, right? So the exoskeleton of a cockroach, and we call them arthropods, right? So exoskeleton of arthropods are made from chitin. That is also type of polysaccharide. Lipids. Lipids is a diverse group of compounds that are hydrophobic, water-fearing, or insoluble in water because they are non-polar molecules. So lipids include fat, oils, waxes, phospholipids, and steroids. So fat, a typical fat or triglyceride consists of glycerol molecule. So here's a glycerol. Glycerol is an organic alcohol. And what attached to glycerol are 3 fatty acids. So here's one fatty acid, being attached through dehydration synthesis, and here we have 3 fatty acids attached. And when we have this fatty acid attached, we call this molecule triglyceride or typical fat. So fatty acid can be saturated or unsaturated. So saturated fatty acid shown over here. So this is a molecule of that has, it's a fat, right? And fatty acid are those tails, and they all are saturated. Those are unsaturated fatty acid. So saturated fatty acids are saturated with hydrogen. The number of hydrogen atom attached to the carbon skeleton is maximized. I know this is a small picture, but if you look at the carbon, each carbon can form 4 covalent bonds, and you can see that each carbon here attached to the maximum number of hydrogen. There is no way you can add more hydrogens over here. So it's saturated with hydrogen. Then hydrocarbon chain contains a double bond, and over here there is some double bond between carbon. Then we have less hydrogen attached, and this is called unsaturated fatty acid. And they have these kinked tails, so they're not straight. And this is the fat that found in oils, and it's better for your health compared to saturated fatty acids. So most unsaturated fats are liquid at room temperature, and they are called oils. Animal fats and meat and the fat contained in butter are example of saturated fat. Another type of fat is trans fat, in the food industry, oils are artificially hydrogenized to make them semi-solid. And when that's done, we create trans fat. So this is a trans fat. You can see this is unsaturated fat because we still have double bond between carbons. So we don't have maximum number of hydrogen. But because hydrogen is located on the opposite side of this double bond, this molecule is still straight. So we just said when these tails are straight, this is not good for our health. Here we have unsaturated fat, but the tail is still straight. So recent studies have shown that increase in trans fats in the human diet may lead to the increase in level of low density lipoprotein or LDL. We call it bad cholesterol, which in turn may lead to plaque deposition in the arteries, resulting in heart disease. Another example of lipid is phospholipid. So this is a phospholipid, and this is a major constituent of the plasma membrane. So here's the plasma membrane, is a phospholipid bilayer, and this phospholipid is a major component of it. And so phospholipids, they have glycerol, right? So this is a glycerol, but instead of 3 fatty acids, we have only 2 fatty acids attached. And in place of another fatty acid, we have this phosphate group. We call it phosphate head, and this is polar head. So it has a charge. And everything that has charge, it loves water. This is hydrophilic part of the molecule, and tails are neutral. They do not have charge, so they're hydrophobic. So structure is glycerol plus 2 fatty acid and phosphate group. One end of a molecule, phosphate and glycerol, is water soluble or hydrophilic, and other end of molecule, 2 fatty acid tails, is water insoluble, hydrophobic. And that's what, and this property allow phospholipids to form this barrier, because hydrophilic part will be facing fluids inside a cell and outside a cell, and those tails will be facing toward each other, away from water. That's how this bilipid barrier is formed. Another example of lipids are steroids and waxes. So steroids have a ring structure. So here we have example of steroids: testosterone, cortisone, cholesterol, and even vitamin D that you will find in your milk. It's also steroid. Although they do not resemble other lipids, they are grouped with them because they also hydrophobic. All steroids have 4 linked carbon rings, and several of them, like cholesterol, have a short tail. So 4 carbon rings and some tails. So cholesterol is a key component of cell membrane, and it's a base steroid from which your body produces other steroids such as estrogen and testosterone. So you can see that's cholesterol, and it can be converted by your body into testosterone in males and estrogen in females. And there is very simple difference, but the small structural difference affect function of these steroid hormones. And waxes like a bee wax or waxy cuticle on the surface of a leaf, it's a coating on the plant leaves that helps prevent them from drying out. So if you see like water is not absorbing by plant leaves, that's because they have this covering that's called waxy cuticle. Okay, you don't need to take this quiz. So we continue. So next, proteins. Okay. One of the most abundant organic molecules in living system are proteins, and they have the most diverse range of function of all macromolecules. So each cell in a living system may contain thousands of different proteins, each with a unique function. They are all polymers of amino acid rearranged in a linear sequence. So here's some example of protein functions. They can be structural proteins, storage protein, contractile protein that your muscles are made from many contractile protein allow you to move. Can be transport protein like hemoglobin in your red blood cells. Enzymes are also proteins. So proteins are made from amino acids. So amino acids are monomers. So when these monomers are put together, we make polymers or proteins, polypeptides and proteins. Each amino acid consists of a central carbon atom bonded to amino group, carboxyl group, and hydrogen atom. So this part of amino acids is the same for any amino acid. What is different is this side group or R group. Every amino acid has another variable atom or group of atoms bonded to the central carbon known as R group. So this different R group make 20 amino acid important for living cells. So enzymes are proteins, function as biological catalysts, and they speed up chemical reaction. And enzymes are not altered or consumed by the reaction. Without enzymes, many biological reactions would not proceed quickly enough to sustain life. So you can see here example of enzyme that is a protein, and then we have reactants. And reactants are also called here substrates. So whatever attaches to enzyme called substrate. And it's attached, enzyme changes its shape, and it's either form chemical bonds or it can break the chemical bonds and releasing product or products, and enzyme is not altered, not consumed, and is available for next reaction. Hormones are also protein, some hormones. We just talk about steroid hormones. Those are in a group of lipids, and most of hormones in your body are proteins. Hormones are chemical signaling molecules, usually proteins or steroids, that's hormones, usually proteins or steroids, secreted by endocrine glands that act to control and regulate specific physiological processes, including growth, development, metabolism, reproduction. So here, what's shown to you on this diagram is a cell, maybe part of some endocrine gland that secrete its hormone in the bloodstream, and through the bloodstream, this hormone can be distributed through your body, and cells that have a special receptors for this hormone, we call them target cells, they will respond to this hormone. So maybe they will start dividing, or they start increasing their metabolic processes, or they start secreting their own product. So it will be some cellular response. For example, insulin is a protein hormone that maintains blood glucose level. So protein structure. Proteins differ in their arrangement of amino acids. So amino acids are building block of a protein. So each of these bubbles represent amino acids. So this is called primary structure of a protein. So this particular protein is made from 129 amino acids. And this specific sequence of amino acid in a protein is its primary structure. So you can imagine if we change this order, or we put more or less amino acid, we will end up with a different protein. Slight change in the primary structure of a protein affects its ability to function. For example, substitution of one amino acid for another in hemoglobin causes sickle cell disease, that is inherited blood disorder. So normal blood cell look like this, like nice kind of like a donut shaped structure without any hole, of course, inside. But people with sickle cell anemia, they have red blood cells, they shape differently, like this, like sickle shape. And this is because most of the structure inside the cell is hemoglobin. And normal hemoglobin, it has this particular sequence of amino acids. So amino acid number 6 is glutamine over here. But when it's substituted with another amino acid, just one change cause a huge difference in the shape of hemoglobin and shape of red blood cells. So this is called mutation. So that just tells us how important this primary structure of the protein. But proteins, they have also the secondary structure. So here's the primary structure, sequence of amino acid. Then this chain of amino acids is folded and rearranged into secondary structure. Two most common are alpha helix, so that's an alpha helix, or beta pleated sheets. So that's a secondary structure, and it's formed because of hydrogen bonds between these, amino acids. Both structures are held in shape by hydrogen bonds. Now, the unique three-dimensional structure of a polypeptide is known as tertiary structure. So over here in a tertiary structure, you can see we have helices and sheets and helices and sheets. So this unique three-dimensional structure is tertiary structure. In nature, some proteins are formed by several polypeptides. So that's one polypeptide. But over here, you can see two polypeptides. Each of them already has a tertiary structure. So when your cells put more than one polypeptide together, the protein it became functional. For some protein, it's important, and it's has its quaternary structure. So in nature, some proteins are formed from several polypeptides, also known as subunits, and the interaction of those subunits forms their quaternary structure. Now, the proteins must have either tertiary structure or quaternary structure, right? This, the secondary and primary structure doesn't allow protein to conduct its function, to carry on its function. So if protein has primary or secondary structure only, they cannot function normally. They must have tertiary structure, and some proteins quaternary structure. Now, when we increase or decrease temperature, or when we increase or decrease pH, this change in the surrounding environment can affect a shape of a protein. So protein shape is sensitive to surrounding environment. So when we have this unfavorable change in temperature and or pH, it causes denature of protein. And denature means proteins loses its shape. So high fever above 104 Fahrenheit in humans can cause some proteins to denature. And on this picture, you see denature of protein in the egg, right? So this white that you have, it's albumin. It's a water-soluble protein. But when you increase the temperature, it changes its shape and it became insoluble. And so it loses its shape and loses its function. And it can be irreversible. Some denature can be reversible, but many of them are irreversible. And the last group of organic molecules are nucleic acids. Nucleic acids are key macromolecules in the continuity of life. They carry the genetic blueprints of a cell and carry instruction for functioning of the cell. And two main types of nucleic acid include DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. So DNA and RNA are the type of nucleic acid, and they involved in protein synthesis. And this is very simple pictures that tell you briefly how proteins are synthesized. So DNA, it's an analogy. They compare DNA with a cookbook. You have many recipes in a cookbook, but you just need one, right? So you kind of photocopy this one recipe through the process of transcription, and messenger RNA is made. That's your recipe. And then ribosomes, those are organelles found inside the cells, is like a chief. And tRNA gonna bring amino acids to the chief, so chief can make protein, right? But tRNA will be like kitchen hand, and amino acid will be like ingredients, right? So to make a protein, that's protein cake, you need information in DNA, and you need messenger RNA, ribosomes, tRNA, and amino acids. DNA and RNA are made up of monomers called nucleotides. So here the picture of a nucleotide. So several nucleotides combined form polynucleotide. So each nucleotide has three parts. It has a sugar, 5-carbon sugar, nitrogenous base, and phosphate group. So in the DNA, sugar and phosphate are the same. Sugar is deoxyribose. In RNA, sugar and phosphate are the same. Sugar is ribose. And what is different are those nitrogenous bases. So each DNA nucleotide has one of 4 possible nitrogenous bases, A, G, T, or C. So adenine, guanine, thymine, and cytosine. So just these bases are different. So that they can be adenine or guanine or thymine or cytosine. So dehydration reaction links nucleotide monomers into a long chain called polynucleotides. And the bond is formed between sugar of one nucleotide and phosphate group of another nucleotide, and so on. And this sugar-phosphate chain is called sugar-phosphate backbone. And what is hanging from them are nitrogenous bases. Now, DNA is a double helix, and this double helix is formed by hydrogen bonds between the bases. The base pairing very specific. So A always pairs with T, and G can only pair with C. So C with G, A with T. Mnemonic is apples in the tree, cars in the garage. All right, and because of this hydrogen bonds, we form double helix. So this is one strand of DNA, right? And when we put 2 strands together and hold them by these hydrogen bonds, that's a double helix. RNA is nucleic acid. It's different from DNA. But first, RNA does not have thymine. It has uracil. So if you see thymine over here, then that's DNA. If you have a uracil, that will be RNA. So, right, so this cannot be RNA. This can be only DNA because it has T. RNA is usually single-stranded. DNA is usually double helix. And of course, sugar is different. Sugar in RNA is ribose, in DNA is deoxyribose. ATP is nucleotide, so it's kind of in a group of nucleic acids, and so it also has a sugar. Sugar is called ribose. Well, here is the ribose sugar, not deoxyribose. It has nitrogenous base, adenine, so it's A. Together it's called adenosine. And instead of 1 phosphate group, ATP has 3 phosphate groups. That's why it's called adenosine triphosphate. And ATP is universal energy source. So bonds between phosphate groups contain potential energy, and breaking the bonds release energy. So here's ATP. When water is added, We can break this bond between the third and the second phosphate group, so right here. And now we don't have a DP, or I'm sorry, a TP, we do have a DP. This is adenosine diphosphate, 2-phosphate, and energy is released, and this energy is used for cellular work. And then this phosphate group can be attached back. Your cells do it, and it's very complicated process called cellular respiration, and we will cover it little bit later. But when this phosphate group is attached, when it's attached back, we form ATP again, we store energy. So ATP is like a rechargeable battery. That's a battery that's full of charge, you can use it, and then it loses its charge, you can recharge it again and start and use it again. So let me see. Yeah, so we have only one slide left. And this slide just a summary of what we cover in this chapter: large biological molecules, carbohydrates, lipids, proteins, nucleic acid. Function: carbohydrates are dietary energy storage, plant structure. Lipids, long-term energy storage like fat, hormones, steroid hormones. Proteins are enzymes, structure proteins for storage, for contraction, for transport, etc. Remember, proteins have most diverse function. Nucleic acid, information storage. Components or building blocks: monosaccharides, glycerol and fatty acids, amino acids for proteins, nucleotides for nucleic acids that made from sugar, phosphate group, and nitrogenous base. An example for carbohydrates are monosaccharides, glucose, fructose, galactose, disaccharides, lactose, sucrose, and polysaccharides, starch, cellulose. For fats, we can have triglycerides, steroids, testosterone, estrogen. Proteins, for example, lactase, that's enzymes, enzyme, hemoglobin, and nucleic acid example are DNA and RNA. Okay, that's it for this lecture. Thank you for watching, and I hope it was helpful.